It's recording. Good evening, everyone. This is Peter Smallage. I'm the New York State Extension Forester. Happy St. Patrick's Day. And I'd like to welcome you to the March 2021 version of the Forest Connect webinar series. Um, as you know, we often have uh, speakers other than me, which is great because it's nice to hear some really fun, cool stuff. And tonight is no different. We're joined by Dr. Paul Jensen. I met Paul when he was working at Cornell, and Paul has probably traveled and worked and studied in a greater geographic span of North America than anybody I know. We were talking at practice time about where all he's been and what he's done. I'll let him fill in the details, but just suffice it to say that I'm very happy that Paul is here. He gave a great presentation at noon, and I'm looking forward to this evening. So, Paul, I'm going to mute my microphone and hover in the background, and it's all yours. Thanks for joining us.
Yeah, thank you, Pete. Appreciate the introduction. And again, happy St. Patrick's Day, everyone. As I mentioned in the first seminar this afternoon, I will fully admit that I am not wearing any green currently. But in this photo of me holding this fisher, I am wearing green pants, so that counts. Um, but I am really glad to be here and excited to share some of what I've learned over the years with you this evening. So I just wanted to start with just a really quick introduction. Currently, I am the Regional Wildlife Manager for New York State DEC Region 5. And I oversee the wildlife unit and really all of our work in the Adirondacks and the Lake Champlain Basin. So that includes a lot of different things from land management on wildlife management areas to protection of threatened and endangered species to game management, including fur bearers and big game, sportsman education. So a whole host of programs that I oversee. And it's just been great. I've been in this position for about 3 years now and have really enjoyed it. But previous to that, I worked for about 15 years, again, for DEC as the regional fur bearer biologist. And so much of what I'm going to share tonight is work that I conducted in that role. And again, particularly in the Adirondacks. Now, prior to working for New York State DEC, I worked for the New York Cooperative Fish and Wildlife Research Unit at Cornell, as Pete mentioned. And I also worked in Alaska for LGL Alaska Research Associates. And we were based in Anchorage. And we did a lot of caribou and other large mammal monitoring on the North Slope of Alaska. And so we did a lot of work pretty much from just east of Barrow to the border with Yukon Territory. So big swath of northern Alaska. And I also did a short stint with the National Park Service in South Florida. So everything from South Florida to Alaska and a little bit of in between. So I think that's what probably Pete was referring to. I received my PhD from McGill University in Quebec, my master's degree from UNH in Durham, and my bachelor's degree from SUNY College of Environmental Science and Forestry. And lastly, I am adjunct faculty with the Department of Environmental Biology at SUNY ESF. So some of the research that I'm going to be talking and sharing with you later in my talk really is part of that appointment that I have with ESF. And I've got a lot of really good collaborators at ESF that I've worked with for many years. So tonight I'm going to talk very broadly about the ecology of American martens in these transitional boreal deciduous forests that we find in Adirondack Park. And just want to kind of give you a brief outline of what I'm going to be talking about tonight. I first want to cover the biology and distribution and some natural history, not a whole lot, but just enough to orient all of you to the two species I'm going to be talking about. Then I'm going to transition into some of the previous research that I've conducted with DEC, and then end with some of the research that I've received funding through the American Wildlife Conservation Foundation, in particular looking at using GPS collars for looking at marten space use. So with that, I'm just going to jump into some of the biology and natural history. This is an American marten. If you look real closely, you're going to see some ear tags. I don't know. Can you, Pete? Can you see my mouse cursor when I move it?
Yes.
Okay. So I'll just use that as a pointer. So you can see some small ear tags here. This was one that I believe this was a female, and she was too small for us to put a radio collar on, so she just got nice earrings. One thing that marten have is this very distinguished throat patch here, this gular patch, which typically is this bright orange, or sometimes it's like a cream color. But American martens are small carnivores in the weasel family or the mustelid family. They generally range from about 1 to 2 pounds. So a female is going to be about a pound, and a male is going to be about 2 pounds. Martens were historically present in all of New York, but the population is now restricted just to the Adirondack Mountains in the northern part of the state. And we can talk, maybe later about what happened there historically. But martens are habitat specialists. They really prefer large contiguous forests, and they also require complex forest structure. So what we're learning, and I'll talk about here in a minute, is that forest composition can be important, but it's really a proxy to that complex forest structure. Now, important prey for martens consists of small mammals, hares, but they'll pretty much eat anything. They'll eat insects and frogs in the summer. They're quite fond of both hard and soft mast. So they will consume beech nuts, mountain ash, apples, blueberries. So really, again, just very wide range as far as their diet. They have a really interesting foraging behavior that they can utilize to sort of capitalize on a prey resource in winter, and that is the small mammals that are below the snow. And so martens have the ability to tunnel down through the snow and exploit that prey base. Martens will typically have their kits in April or May, and litter size is about 2 to 3. And their home ranges are among the largest for mammals per unit body mass in North America. And so a home range of a male is going to be about 2 to 6 square miles. And for a female, it's about 1 to 2 square miles. Now, predators include really quite a few species of fishers, coyotes, fox, bobcat, and birds of prey. When we were doing our study years ago, we were radio collaring animals, we had really a couple of main predators in our study area, and that were fishers, and we had one that was preyed on by a great horned owl, and that was kind of interesting because we actually could see the whole kind of record of what happened in the snow and found a feather. Now, whenever I talk about martens, I have to talk about fishers too, because they're really ecologically similar to martens, and they engage with them in what's called intraguild predation. And that is a combination of both competition and predation. And again, we'll get into some of those details here in a minute. But fishers occur statewide in New York State, and they're wide ranging throughout the northeastern US, and even down into some of the Appalachian states like West Virginia and Pennsylvania. They're quite a bit larger than martens. A male is going to weigh up to 15 pounds, and again, they're in the weasel family. Now, one of the things that folks used to think is that fishers were more of a habitat specialist. And so what we're finding, though, is that they're really more of a generalist than what we previously thought. So whereas martens really are associated with contiguous forest, fissures are much more apt to occur in landscapes where there's more heterogeneity. So you can envision, an area where you have forested land interspersed with agricultural lands, and even sometimes suburban areas. So we have one of our areas to the south in Saratoga County has probably one of the most robust fisher populations in the state. And it's very much suburban areas and kind of mixed with natural areas. And so again, much more of a generalist than we initially thought. Prey consists of small mammals and hares, squirrels. They're very keyed in on white-tailed deer, dead white-tailed deer or carrion in the wintertime. So they exploit that. They're scavengers, and martens scavenge as well. But they will also kill porcupines, one of the few predators that can kill a porcupine, which they do by just repeated bites to the head, and very quick repeated bites. Now, fishers forage primarily above the snow, so they really don't have that ability to exploit that subnivean zone, and it's because they got a larger body mass, right? They have a similar body shape, but much larger, and so really they can't get into those real tight, small spaces. They have a very high dietary overlap with coyotes, and they've got similar reproduction as martens, on average about 2 to 3 kits, born mostly in March. And again, some very large home ranges, about 6 square miles for a female and 12 to 15 square miles for a male. Now, predators primarily include bobcats and coyotes, but we also see some birds of prey that will take fishers. Primarily bald eagles are notorious for that. Now, both martens and fishers occur over much of the boreal region of North America, where they're sympatric, and you can see that in this large gray swath here. And much of the historical distribution of martens and fishers are, they're pretty much intact. A little bit less so for martens, especially in the Northeast and in the Midwest. But if you kind of like hone in here on New York, you can see that our population in New York State is geographically isolated. There's no connections with other marten populations either in New England or in Southern Canada. So kind of what we have is what we have. And so there's certainly some important conservation implications associated with that. So the other thing I just wanted to point out with this map is that in general, you see that martens occur really in one of two situations. So here in this map, marten is represented by the green, where they're sympatric with fishers, and then the blue, where they're allopatric, where they occur by themselves and fishers don't occur there. So you see them occurring in one of two situations. Either number one, it's at high latitudes in that boreal region, or it's at higher elevations in those lower latitudes. So if you're in the lower 48, most of those marten populations, with the exception of probably Minnesota and Maine, are in higher elevation areas. So in the west, that includes mountain ranges like the Rockies and the Cascades and the Sierra Nevadas. And in the east, you see that in the Adirondack Mountains, as well as the northern Appalachians in Vermont, small portion of Vermont and northern New Hampshire, and also in portions of Maine. So across this range I just showed you, martens really use a variety of forest stand types, and I mentioned that previously. But really kind of like the paradigm for quite a while was this thinking that marten habitat, or martens required these old growth coniferous habitats. But new research has really shown that they'll use a variety of stand type. And really what is more important than forest composition is the actual structural attributes of the stands. So these are some typical forested areas that you might see martens in New York State, and probably representative of many of the habitats in the Northeast. So they include coniferous stands, mixed stands that really have higher proportion of coniferous stands within these riparian areas, wetlands, and other drainages, but for the most part are dominated by deciduous forests. And you may be surprised to learn that martens also use deciduous dominated stands. And some of the adult martens that we've radio-collared in the past have had home ranges that were really dominated by deciduous forests. Some had up to 95% deciduous forest within their home range. But again, really the more important feature of marten habitat is not so much related to forest stand composition, but the structural complexity of those stands. And so martens, really require structurally complex forests for literally all aspects of their life history. Denning and kit rearing, thermoregulation, foraging, escaping predators, and resting. And so when we look at sort of the structure, we can think about it in a couple of different ways. One is the vertical structural complexity, represented by stand density and to some degree the composition and the diversity of tree species. But this, for example, would be some place that this time of year you might radio track a marten and find it during midday resting in the canopy. It might be up on a branch like this here, just kind of laying. And you can imagine that this is a pretty safe area for a marten to rest, right? For a couple of reasons. Number one, you can imagine it being very difficult for an avian predator to navigate this type of complexity without the marten being alerted to its presence and being able to escape pretty quickly. Secondly, they require this structure that allows them to move across the canopy easily. And so you can imagine, okay, here's this marten up here resting again, and a fisher comes up this tree. If this tree is out somewhere by itself, that marten really has nowhere to go. It either has to try to bypass that fisher by coming down a different side or jumping. Not likely going to be a good outcome for that animal. However, in a more well-stocked stand with closed canopy, that marten can escape across that canopy. And I was sharing with the group, the last group, that it's amazing how quickly martens will navigate the canopy. And I've seen martens move across the canopy just as quickly as they do on the ground. So they're very, very adept at moving across that canopy, and that closed canopy really is super important for escape cover and protection from avian predators as well. Now, the other important feature of this complexity is a horizontal complexity, and that's represented by coarse woody debris on the forest floor. It could be wind throw, it can be large diameter trees that have fallen down. But this is really, really important structure for a couple of reasons. First of all, their primary prey, small mammals, are closely related to coarse woody debris themselves. And so in the wintertime, this would be completely covered with snow, but there would be air spaces underneath this coarse woody debris where those small mammals are overwintering. And so again, martens have this ability to find breaks in the snow and tunnel down underneath and exploit these subnivean spaces and find small mammal prey. Again, something that really, with the exception of weasels, other carnivores just do not do. Fishers, again, will do it to some extent, but it's not their primary foraging behavior. So we've talked about some of the predators of these two species. So let's just talk a little bit more about some of the community interactions that occur. And in the Adirondacks, and I would say probably in a lot of areas of the northeastern US, the most abundant members of the predator guild includes coyotes, fishers, and martens. And there's other species as well, but they certainly don't occur at the abundance of these predators. So those species include bobcat, fox, and probably to a lesser extent weasels. We don't really have a lot of information on weasels. But all of these carnivores here compete for a similar prey base. And I'd mentioned some of these things before. Things like snowshoe hares and deer carrion are super important for fishers and coyotes. Hares are also really important for martens. And then there's the small mammals, species like squirrels and redback voles, and probably to a lesser extent shrews, but all exploiting this common prey base. So not only are these carnivores competing for this common prey base, but they're also interacting with one another. And that's represented by these arrows. So we have coyotes that will kill martens, as well as fishers killing martens, bobcats killing fishers, coyotes killing fox, fisher killing fox. So a lot going on here in this community. And so sometimes those interactions include direct killing of one species by the other one, by the dominant predator. And other times they create what is known as a landscape of fear. And a landscape of fear really is something that elicits a behavioral response by the inferior predator. And that behavioral response has a fitness consequence. So you can think about a situation where one species may be feeding on a carcass and it's not the dominant predator. And so it has to spend time. It can't spend all of its time feeding. It also has to remain vigilant because it knows, it's aware of its community, and it knows that there's these other predators that are there. And so there's this landscape of fear that gets created that has fitness consequences to it. Both martens and fishers and their prey are also influenced by mast crops. And we had mentioned these again, beech nuts and mountain ash, as well as cone crops. Now, cone crops really influence small mammals directly, not so much the predators. But again, fishers and martens will directly consume soft and hard mast. And one of the early diet studies that my predecessor conducted with fishers is he was opening up fisher stomachs, and it was a big beech nut year, and the stomachs were just completely packed with beech nuts. So it's a really high energy, easily obtained food source that doesn't bite and fight back. So again, they'll definitely exploit this food source when they can. Now, lastly, I just wanted to mention the role of snow, in that snow can mediate some of these interactions among these carnivores. And so, for example, for martens, interactions with fishers are mediated by deep snow, because again, martens are able to exploit that small mammal prey base in the subnivean zone, where fishers really can't do that. So it's in these deep snow, low productivity environments where martens become the superior competitor. And so they occur at higher densities than fishers, and they're able to live in areas where fishers really can't. And that, that relates back to that slide that I showed you where I talked about martens either occurring in high latitudes or in high elevations in lower latitudes, because typically these are low productivity, harsh abiotic conditions. And those are the types of conditions that marten really specialize on. So I'm going to play this video clip. I've talked a lot about subnivean foraging. I just want you to see this short video. Stacy McNulty from the Adirondack Ecological Center and I worked with BBC on this. And some of this footage is directly from Huntington Forest. Other in the introduction is not, but some pretty amazing footage, and I hope you enjoy it.
They begin where the trees begin, in the far north. This is the largest forest of all, an unbroken band of coniferous trees that wraps around the globe, the Boreal Forest. In winter, it can appear empty. But this endless maze is the arena for a unique game of hide and seek. Snow falls in this forest for months on end. As it collects on the fallen branches and trunks that litter the forest floor, a hidden labyrinth of snow tunnels is created beneath. Here, out of the icy air, is where the forest's rodents spend their winter. Prey exists even in this seemingly empty forest, if you know where to find it. The American marten. It only lives where in winter the snow is deep enough to create this maze. Its slim body is ideally shaped for searching snow tunnels. But that shape means it also loses heat quickly. To stay alive, it must continually search for food. The marten picks up a scent. It listens for any movement. Found you. One mouse is not enough.
Okay, well, I hope you enjoyed that. That was like I said, a really cool project to be involved with, and we were just blown away when we watched it for the first time. So, if you're interested in watching the full clip, it's the full documentary is called The Hunt, and produced by BBC. So I think you can find that in wherever you can stream it. So at this point, I now want to kind of transition and really focus on some of the work that I've done with New York State DEC, much of which, not all of which, has been funded through the Federal Aid and Wildlife Restoration Program through the federal government. And really, most of my research has really focused on resolving uncertainty in spatial and temporal drivers of marten populations. And so really I've been kind of interested in some of the basic questions that ecologists ask about the distribution and abundance of species. And as a fur bearer biologist, I've also really have kind of focused on using sort of a combination of harvest data and field-based methods to answer questions. And so our harvest monitoring for martens and fishers really is integrated into our research, and it informs our research. And that's something that I think you'll see as we progress through the rest of the talk tonight. So one of the first things, as a biologist, I was really interested in, okay, where do martens occur in the Adirondacks? And at that time, we really were just kind of coming into the age of GIS and some of the other technologies where we could actually map locations. But it was interesting because I found some of these old maps, and the base map that you see here is probably from about the 1960s. I haven't been able to date it, but it shows you just how restricted our population was at one point, where that red area really indicates that population core, and then the green and blue is sort of the peripheral area, but really kind of focused on the high peaks and constrained to the high peaks with just some residual populations to the south and to the north. So I obviously wanted to get a better and more fuller understanding of marten distribution. And so as part of the harvest monitoring program that I started to develop in 2005, sort of implemented this integrated system. And what this did was, again, it was collecting data from fur trappers, and it linked trapper specific data. So for example, harvest locations, trapping effort, trapping behavior, for example, what species they're targeting. So trapper specific data with biological data, such as sex and age, because we required trappers to submit carcasses of the animals that they trapped. And so the trapper data and the biological data were linked with these spatially explicit harvest locations. So trappers would mark on a map, on a really highly accurate map with a lot of geographic reference points, would mark locations and then would provide information on each one of those animals, as well as providing data on effort and submitting the carcass. So these data have really been useful not only for monitoring harvest over time, but also answering ecological questions. So in addition to these locations that you see here, I entered historical harvest data as well into a GIS database. And again, you can see this really provided us with a much better understanding of marten distribution in the Adirondacks. So all in all, it resulted in about 3,300 locations, and about 2,000 of these records also had the trapper and the biological data associated with them. So as you can see, the vast majority of locations are located along a southwest to northeast orientation. And really the data have really shed a lot of light on, again, on marten distribution. And so some of the questions that we've been able to ask beyond just distribution is why do they seem to be oriented in this southwest to northeast orientation? And why are martens constrained to this area of the landscape? Sure, we do have martens that occur in other parts of the Adirondacks, and even some that occur outside of the actual Adirondack Park. But for the most part, they're very much clustered in the central Adirondacks. Excuse me. So one of the hypotheses that we were particularly interested in testing was developed by Bill Crone from Maine, from the University of Maine in the mid-1990s. And so he posited that martens are limited by fissures, and fissures are limited by deep snow. And so this was sort of a logical hypothesis for us to test, given that this distribution aligns with the highest elevations of the Adirondacks, and presumably in an area with deeper snowpacks. And so we've also collected similar data on fissures, and we've used these data to model habitat selection of both species and also understand marten range expansion over time. And so the next couple of slides will show you the model outputs from these resource selection probability functions. So again, they're really kind of looking at habitat selection. These models are focused on habitat selection at this larger scale. And so from this, you can see that fissure probability of use is really highest in the peripheral areas of the Adirondacks and also in Tug Hill, and lowest in that central region where martens are distributed. And so in some of the other influential covariates in the model included March temperature. So fissures were positively related to March temperatures, positively related to snowpack, as well as the percentage of coniferous stands in the area. And they were also negatively related to coyote abundance. So some of the things that we kind of thought might be going on actually were going on as far as these interactions with other carnivores. Now, when we looked at our marten resource selection function, we really saw kind of the opposite pattern. Highest probabilities in the central Adirondacks, again, pretty closely aligning with that distribution, and then much lower probabilities in the peripheral areas as well as the Tug Hill. And so what we did, one of the big reasons why we developed that fissure model is because we wanted to use the probability of fissure use as a predictor in our marten models. And in fact, that was one of the most influential covariates in the model. But coyote abundance also appeared as an influential predictor. So The other thing that was really interesting, and before I leave this slide, I'll just mention that we did publish this work in the Journal of Animal Ecology. So if it's something that you want to kind of read more about, you can just Google my name and Journal of Animal Ecology, and that paper will come up. But what I was saying was the other really cool thing about the data was that because we had spatially explicit age data associated with these harvest locations, we were able to look at the age structure of the population relative to the core range of marten, and we learned some interesting things. So first of all, just to orient you, the black circles are adult martens, those are harvest locations, so adult martens there, and the red dots are juvenile martens. And so if you look at the area that we predict martens to occur in, and again, this is where they're sympatric with fishers, we see that there's about 0.7 juveniles per every adult. Now, if you compare that to the area outside, you see that it's got a much higher juvenile to adult ratio, almost 2 to 1. And so what's happening here is that martens are successfully dispersing across this landscape into these peripheral areas. But because this represents a time series from 2005 to 2015, we're not seeing adult martens being harvested in this area. So if you couple that with the fact that when you look at the relative abundance of fishers and you compare that in the area where martens and fishers are sympatric, that ratio, this harvest ratio is about 1 to 1. So fisher trappers on average, or excuse me, trappers on average are taking about 1 fisher for every marten. Now, that's a little bit biased because there's no bag limit on fisher, but there's a 6 animal bag limit on marten. So it's really, that's probably not very accurate, but the bigger picture is looking at how does that change when you come outside into this peripheral area. And so that harvest ratio in this region here is 39 fisher for every 1 marten. So very large differences in relative abundance of martens and fishers between this area that we identified as this core region and this peripheral region. So we're in a situation where we have juvenile martens that are dispersing outside this core, and this population continues to expand. But right now, we have abundant fisher and coyote populations that exist in this periphery. So these juvenile martens are dispersing, but they're not surviving. They're getting trapped, but if they were surviving into adulthood, we would have many more adult martens being taken in this region. So now I want to shift gears, talked about some of the spatial ecology work. I want to shift gears to looking at some of the temporal changes in marten populations. And we'll come back to some of the spatial ecology. But this was some work that really, it involved radio collaring martens, but even before we started doing that work, again, we really started to drill down on some of the harvest data and look at some of these interesting patterns that we were seeing. And what we were seeing were these really wide fluctuations in trapper harvest success rate. And so, again, as a new fur bearer biologist, I'm looking at these data and it's a real head scratcher in terms of, okay, what in the world is going on here? So I was challenged by, some others in the agency saying, well, maybe that's somehow the result of trappers that are over harvesting these animals and there's all this variability. I didn't quite buy that. But some immediate observations that we had were that, number one, that marten and fisher harvest rates in Adirondack Park were in sync, and they fluctuated in this alternate year pattern. So in one year, harvest success rates were really low. The next year, they were very high. And that was different. So, if you look here, this line here is marten. This solid line up here is fisher harvest. But fishers outside of the Adirondack ecoregion were doing something completely different. They weren't showing this high level of variability from year to year, and sort of an increasing trend in harvest success rate. So really trying to understand what in the world is driving these fluctuations. So I started working with a fur bearer biologist in Maine, Wally Jacobus, some of you may know him, as well as a few colleagues at the, excuse me, at the Adirondack Ecological Center. And so we began to really try to drill down on what was driving these patterns. And interestingly, it was a trapper from the state of Maine that was talking with Wally at one point that had, literally experienced these same types of fluctuations on his trap line. And he made the comment, hey, you know what? I think that they're somehow related to these cycles that we see in beech nut production. And that really kind of is what launched this research, where then I kind of started partnering with biologists at the Ecological Center, and we started to bring data to bear on this question. So when we looked at their mass production data, which they had been collecting for several years back to 1988, we started to see some of these very familiar alternate year cycles that, again, were apparent in the harvest rates. And the harvest rates had a strong inverse relationship with mass production. So again, there were high harvest rates in mass failure years and low and approaching zero when there was large mass crops. So the other interesting thing that we found was that many of these mast and seed producing trees are synchronized, both among species and over large geographic extents. So here you can see a lot of these species will fluctuate in a similar manner. They might not have as strong of a pattern as American beech, but in particular, American beech and mountain ash were highly correlated. So they pretty much fluctuated exactly the same each year. And again, these are really important food sources for both martens and fishers and their prey. So there's sort of a double benefit for these predators. They can eat the mast directly, and then they can benefit from the small mammals that are benefited from that food source. And this is really especially important in the central Adirondacks, where the basal area of beech reaches its maximum across its range in North America. So this area here that you see that reaches these maximum basal areas, this also coincides with that core marten range that we've identified. So this is really, like, you can think of this as just a huge and vital source of energy that pulses and flows throughout the entire ecosystem. And so it has really large impacts on both prey and predator populations. And again, we see this when we look at the small mammal data collected at the Adirondack Ecological Center. We see these same patterns where for several small mammal species like redback voles and short-tailed shrews, we see these peaks and these valleys that correspond to those mass crops. Now, small mammal relative abundance, these graphs that you see here, were collected using live trapping data during the summer. So what happens is you get these big mass crops that occur in the fall, and then there's a lag numerical response by the small mammals. And that happens because those mass crops, again, are important food sources for those small mammals. And what they do is they increase overwinter survival, and they likely also result in earlier breeding in the spring. So they start breeding sooner, and that allows them to also have more litters. So if it's a big beech nut crop in the fall, my recommendation to you is to buy as many snap traps that you can, because you are going to have a lot of mice in your house in the middle of summer that following summer. And it's interesting, and we've seen it in the Adirondacks, and I'm sure you've seen it in other parts of New England, where hardware stores will actually sell out of mouse traps. And this is the dynamic that's going on. So in a nutshell, what we learned from this research is that that annual variation in harvest rates was best explained by an immediate response of martens to autumn mass crops. So them either directly feeding on that mass crop or that mass crop not being available for food. Now, to a lesser degree, the relative abundance of small mammals in the summer after a massing event also explains some of this variation. And so we observed a notable increase in the juvenile cohort of martens in the autumn after a masting event. So this is called the silver spoon effect. So what happens again, just to kind of follow the timeline, is big masting event in the fall, followed by a large population of small mammals in the summer. And those small mammals are available to the kits of martens and fishers during the summer. So you have higher survival rates of those kits. And then this large cohort of juveniles then moves into that next autumn. And because this is an alternate year cycle, that big cohort is then moving into a time when the bottom is going to drop out of the food source, because that's a mass failure year. So together, those two things are really is what's driving these harvest rates as a function of both the capture probability of martens, which is likely due to influences on space use and movements, plus the numerical responses to prey availability. So the next part of this was sort of taking what we learned from these harvest data, and then sort of moving that forward into a field investigation. And so saying, okay, we hypothesize that really what's going on here is that during a mass failure year, there's little food that's available. And so these animals are moving more, they're coming into contact with traps more often. And so that's really what's driving these capture probabilities. And so we designed some research to look at these questions and really understand how these post-mass crops were influencing both marten space use and resource selection. So we carried out the field portion of this work in the central Adirondacks, again in that core region, at 2 study areas up here in the High Peaks region. This is Huntington Wildlife Forest at the Adirondack Ecological Center. This is Elk Lake Clear Pond Preserve. And this is the West Canada Lakes Wilderness area down here. So just real quickly, methods, we live trapped individuals. In our 5 field seasons, we captured over 100 martens. We aged them, sexed them, put ear tags in, and then we also radio collared them with these conventional VHF tags. Some of you may be wondering, I forgot to mention this during my first talk, but some of you may be wondering what we use for bait. And you can see the sardine can right here. We use sardines just out of convenience, but we also put jelly on top of the sardines. And many times the martens would eat the jelly and they wouldn't touch the sardines. So, and I can't say I blame them there, but my technicians seemed to like the sardines, and I oftentimes would find them sneaking sardines in the field. So with that, many times we would process these animals if it was fall or summer, which is a great time to do field work. We could process these animals on the back of a truck and a truck bed. Wintertime was certainly more difficult and usually, involved the use of snowmobiles and trying to keep the animals warm. And so, but we were able to, again, collar quite a few individuals and then track them using aerial telemetry techniques, either using a Cessna 172 or small aircraft. And that really was our primary means of relocating these animals on the ground. It's a very tedious process. And so, we would go up for, say, anywhere from 2 to 3.5 hours and locate these animals. And you have to do a lot of circling. There's antennas that are mounted on the wing struts, as you see here, on both sides of the plane. And so you can either have both antennas on or switch one on and the other one off. And by doing that, you can winnow down where that animal is on the ground. And so it's pretty expensive. You're talking $150 to $175 an hour. Of course, my day is taken up doing this work. And again, you're only collecting 1 point on an animal per flight. So you're going up, collecting a location on, say, anywhere from 8 to 12 individuals, and then maybe going, getting back up in the air a couple of days later, having perhaps 3 flights a week at most. And so you're getting the sense that the collection of these data is very tedious and it's very expensive. And I'm going to contrast that with what we've done with our GPS collars. This just shows you a map of some of the home ranges that we were able to generate at Elk Lake. Some examples. You can see on the left, the green, blue, and orange were males, and the pink is a female. And this is something I failed to mention at the beginning, is that martens are intersexually territorial. And so what that means is that males won't overlap with other males, females don't overlap with other females, but as you can clearly see here, they will overlap one sex over the other, so that female home range that you see is overlapping 3 male home ranges. So I'll say it later on, but really for the most part, we had hoped, because again, we were trying to look at the influence of mass crops on an annual basis and how that influenced space use. We were really trying to estimate seasonal home ranges. So autumn, winter home ranges, and looking again at how that mass influenced the use of that space on the landscape. However, I wasn't able to collect as much data as I'd hoped. And so by and large, I really was relegated to estimating aggregate marten home ranges over, over a year, or in some cases over multiple years, and just pooling all the data. And then you can see some of the home range estimates in that table at the bottom, using a couple of different estimators. The important thing there too is to look at the size differences in space use. So a male, that last column, that male to female ratio, that's the ratio of the area of a male home range to a female home range. So for both estimators, it was about a male's home range was about 2 times as large as a female. And that's consistent with the literature and what others have found. We are also able to do some kind of comparison of space use relative to mass production, because we did have sufficient data to compare paired estimates for 5 individuals. So these were individuals that I had enough data to generate home ranges for a massed year and a mass failure year. And so what I found was that for those paired estimates, home ranges were about 2 to 3.5 times larger during mass failures than when mass was produced. And then when I looked at, sort of not really concerned about pairing them, just looking at comparing all home range estimates for mass failures versus mass produced, I increased my sample size a little bit and found similar results that, again, that those home ranges were about twice as large during mass failures. I mentioned that third bullet point. And lastly, we found that yes, that we confirmed that hypothesis that space use is likely driving capture probabilities. And so what we found was that those animals were really moving more within their home range in those mass failure years than when mass was produced. And so that was really a big driver of their capture probability by fur trappers. However, again, sort of somewhat foiled by sample sizes, I always thought, and I would often talk with our technicians and say, man, wouldn't it be great if we could use a GPS collar on martens? And certainly they're available for these larger mammals like moose and wolves, but really even today, there's not a lot on the market that's commercially available to use for these small mammals, these GPS collars. They're just kind of coming online now. But that leads me to my last portion of my talk, which really is, again, is moving along this vein of understanding movements and space use of martens relative to these pulses in mass production and prey fluctuations. And I've been working with my co-PI, Stacy McNulty, at the Adirondack Ecological Center and ESF. And we're really, we've been able to advance this work greatly through funding by the American Wildlife Conservation Foundation. And that allowed us to do some initial testing of these tags, constructing these GPS collars, and then actually deploying them in the field. And so that's what I want to talk about here in the next few slides. So the first thing I wanted to do is to see how well do these tags work. You can see in that right-hand corner how small they are. This is less than a 10 gram GPS tag. There's the chipboard that you can see there, and then mounted on top of the chipboard is the antenna, the patch antenna. And so we deployed these tags just like this. Usually I put them in just a clear Ziploc bag. We deployed these tags during autumn of 2018 in 3 different forest stand types with heavy canopy cover, as well as a reference cover, a mowed field. And really we're interested in a couple of different metrics. One was estimated fixed success rate. So that was simply, if I programmed the collar to attempt 8 fixes in a day, what proportion of those were successful? We also were interested in location error. And so that was determined by taking a professional grade GPS unit and letting it average many, many points and getting a really good location on where I was placing that GPS tag. And then allowing that GPS tag to collect data over time, and then comparing the estimated location with the quote unquote true location. And then we also looked at what's called the circular error probabilities. So that's looking at what error is there at different probability levels. So in other words, the 50% circular error probability might have error that ranges from 10 meters to 30 meters, and then it would go out from there. So that's what that circular error probability is all about. And so this is a table here from our results, but I kind of summarized it for this presentation. We collected over 10,000 locations. We saw a fixed success rate of between 61 and 86%, which I thought was really good. Location error between 19 and 49 meters. And then you can see the circular error probabilities of what those captured. So for example, again, 50% of the locations were between 15 and 37 meters. Now, some of you may think, well, especially if you've worked with GPS collars before, you may think that's not good at all compared to some of these larger collars that are used with moose or other large mammals. But you have to realize that number one is that when we do aerial telemetry, almost always, no matter where you are, that error is going to be at least 100 meters, and in many cases greater than that. And so what we recorded in mountainous terrain in the Adirondacks was anywhere from about 250 to 350 meters. So these GPS tags are providing a much finer resolution than what we could get with the aerial telemetry. Not to mention that these tags are actually pretty cheap. I can construct a collar for about $75, which is about a half an hour of flight time. So the capability to collect a lot of high quality data is it's just head and shoulders above what we can do with the VHF collars. So the next step in all of this was, okay, I've got a tag that I think is going to work well. And so how do I integrate this into a collar that can actually be deployed on an animal? And so this was version 1. There's the tag at the top, and these wires are the location where this tag would be mounted in a horizontal position. And so we use a lot of epoxy here to try to protect the battery, and we never deployed this. It just was too clunky. We didn't have a nice circular collar. It was too heavy. We could only deploy it on males anyway. But overall, it just was just not a good fit, but at least a good start. And so from there, we went to version 2. This is where the engineer that I was working with at Perthold Engineering obtained a 3D printer, and he was able to place both the battery, which is here, as well as the GPS chip and antenna within these really hard plastic boxes, and then connect the two via wires. Now, one of the things I mentioned, again, during my last seminar, is that one of the real tricky things with developing a collar like this is that you have to have this counterbalance perfectly, because what you're attempting to do is keep this GPS antenna at as close to the noon position as you possibly can, because that's where it's going to get the best reception and be able to collect the best data. So you wouldn't want a situation where this component here was heavier than the VHF tag, because of one, then what would happen is that the center of mass, this would shift, right? And this would be on the bottom pointing to the ground. And I did that test and found that the fixed success rate was 0% if that happened. So the counterbalance was a key issue. This was a design that I liked and I still probably will use. The only problem with this design is that you need to use a heavier VHF tag. And so that really precludes using this collar on a female. Now, one of the things that you're not seeing here is that the other thing that we do is that we put really heavy duty shrink tubing over these components just to add another level of protection. But this is the design that we're currently using, the one that we kind of landed on. And this is a design where that GPS antenna is mounted directly at the noon position. The battery is mounted at the 6 o'clock position. And the wires go between the two. Again, you can see that shrink tubing. Now, this collar here is light enough to deploy on a female. However, what you'll see being absent is the VHF tag. Certainly doesn't have that big VHF tag that I showed you in the previous slide, but it doesn't even have the smaller tag, which this smaller tag was really instrumental in shaving down some of the weight as well. And so this is actually constructed for avian use, for avian research. And so, of course, with birds, you need very, very light tags. So I was able to obtain some of these tags, and you can see in this male collar here, I mounted that, epoxied that VHF tag on the side. And the one really cool thing about these collars is that they're rechargeable. So you can reuse them over and over and over. Now they have a fairly short life. So for example, if I was trying to collect 6 to 8 locations a day, that collar is only going to last for about 3 months. So it necessitates going back out into the field, recapturing the animals and recollaring them, which for martens, that's not a huge deal because you can recapture these individuals pretty easily. But it does require having, you know, a pretty good supply of these tags on hand that you can swap out. So I'm sure, you know, we'll continue to make some adjustments to these collars and tweak them, but I think we're pretty close to a final design. And then we actually took them out and we deployed them. So we deployed GPS collars on 3 adult males. I haven't had a chance yet to deploy that smaller collar on the female, but again, these were 3-month deployments. And we had a single deployment on 1 male at Huntington Forest, and on a male at Elk Lake, we had 2 deployments, and on another male, we had 3 deployments. So able to look at our fixed success rate. Wasn't quite as high as what we saw with the static test, but it was approaching it. So about 40 to 60%. However, we did have one deployment where something went wonky and we only got 25%. So I don't know if the collar, you know, somehow rotated perhaps, and the orientation of the antenna was not ideal. But on that one, we did get a lower success rate. Then we had one that slipped the collar after 3 weeks, but in that 3-week period, we collected 105 locations. On the 2 other males at Elk Lake, we collected over 500 locations. So this is just, you know, an order of magnitude above what we could do with VHF tags at just a fraction of the cost. And we did have a single mortality at Elk Lake in May, and I'm fairly confident that that was a fissure that killed that marten because the side of its skull was crushed in, and you could see the you know, the outline of the canine teeth, and they were spaced what you would expect for a fissure. And these next 2 slides just show you some of what we've been able to collect during our field deployments. You can see here the locations at Huntington Forest for this male, and then these two males here. And then just kind of zooming in on that, you do pick up sort of these interesting movements that animals make sometimes. So they have these home ranges that they defend their territories, but they do make these these jaunts out into the outer reaches and then come back into their home range. This marten here had an interesting home range configuration that I've never seen before, in that it's highly linear, partly probably due because of this river here, and could be maybe partly due to the clear pond here, and maybe there's some other martens in here that are forcing it to sort of have this highly linear home range. But here again, it just shows you how these two males are not overlapping. So they're maintaining exclusive territories. But in summary, overall, I've been really impressed with the performance of these collars, and I believe that they have really a lot of potential to improve our understanding of marten ecology, and in particular, help us to better understand how space use and movements are influenced by these pulses in mass production that we see. And so this funding that I received through the American Wildlife Conservation Foundation was really instrumental in doing this pilot phase and getting all the kinks worked out of these collars, and then actually going out in the field and doing these test deployments. And then lastly, the other thing, coming back to this slide, coming back full circle, is we've also been able to use this technology on our fisher collars. And we're really, with this study that we're conducting now, we're entering our fourth year, this upcoming field season. And we are focused on adult females. And so all the adult females are getting commercially available collars that are very expensive. But on the adult males, we are using these collars that we've developed for martens, and we're deploying them on fishers as well. And so that's really going to greatly expand our ability to collect data on movements and space use of these animals. So with that, again, I just wanted to thank the American Wildlife Conservation Foundation. If you've never had a chance to peruse their website, it's awcf1911.org. And they're really involved with a lot of interesting work, not only in New York State, but across North America. So I really encourage you to go there and check them out. And then lastly, just a ton of people over the years. This is just a fraction of the people that I've worked with and have assisted me. And a lot of landowners that have allowed me to have the privilege of going on their lands and conducting this research. And lastly, as I said, this work's mostly been funded through Federal Aid and Wildlife Restoration Program. And also you saw the work that the American Wildlife Conservation Foundation has funded. So with that, I'm going to end, and I'm also going to just put up this report and this reference here, because in the last talk, I had quite a few questions on timber harvesting and harvesting prescriptions and what do martens require. And that's really not within my wheelhouse. Dan Harrison at the University of Maine has done a tremendous amount of work in this area with many students over the years. And so he's certainly a good source of information in the papers that he's published and the reports. And this report here is a good report to just kind of get a general overview of marten habitat requirements and just, again, some very general prescriptions for timber harvesting. And so I would just encourage you, and this may take a little heat off of me of trying to answer questions that I'm really not too qualified to answer. So with that, I thank you. I hope you enjoyed the presentation and look forward to hearing your questions.
Great job, Paul. And you did a very wonderful job at lunch answering forestry questions. So, but I will try and find that find a link to that reference, and then I can share that around. So there have been several questions. It's a very engaged evening audience. Sometimes the evening audiences are like they've had a long day, you can tell, but this group is very interested in the topic. So do you want me to read the questions to you again, Paul? Hello, hello.
Sorry about that. I had myself muted.
That's okay. Oh, I was here like, oh no, what button did I click?
The classic Zoom mistake.
Yeah, that's every right.
Yeah, what I said what I said was I can see the chat, except I would probably have to scroll up. So if you've got the questions queued up, that might be the easiest way to do it again.
Sure. Yep, sure.
Okay.
Okay, so first question from Mike wants to know if turkeys and grouse are also prey items for martens.
Well, turkey and grouse, certainly grouse in the winter for marten. And we've done a lot of tracking with our collared individuals, backtracking them and looking at sort of their fine scale movements in the snow. And there were I wouldn't say several, but on a few occasions we found kill sites, and a few of those were in fact ruffed grouse, and so you just see a big pile of feathers. So yeah, they do take them, and I do wonder if martens get them when they're in their snow roost. And so many of you may know that in really cold temperatures in the winter, grouse will sort of burrow into the snow and have that like an insulative blanket around them, and they'll be sort of with their head tucked down and in this snow roost. And I've often wondered if martens detect those grouse and smell them or hear them, and then very closely, get very close approach and then, pounce on them. Now, turkeys, I've never really heard. I'm sure a marten would take both eggs and the poults. And fishers, I think, too, also have a very bad rap with turkeys. But some of the papers I've read on fisher diets really suggest that that turkeys are a really small proportion of the diet.
Okay. And there was in the noon hour, somebody found a link for the BBC video that you referenced, and so I pasted that into the chat window, and I can share that with you, Paul, in case you want me to, in case you're interested.
Okay, yeah, good.
So Gary wants to know how the pelt values have changed over time. That was first question.
Yeah, so pelt values do change pretty dramatically. And I will say in general, the fur market has been depressed for a good number of years. I want to say, it's been a while since I've acted in the capacity of fur bearer biologist, but I want to say it's probably going on close to 10 years of a fairly depressed fur market. Now, marten in North America are like the bread and butter of the fur trade. It's a major commodity that is exported from both the U.S. and Canada, and so interestingly, many times in a depressed fur market, proportionally, other pelts will decrease dramatically, but marten pelts won't decrease to that same level. So they do have this ability to sort of weather these changes in demand and fur market price. But so I can't really tell you. I can tell you that when I was the fur bearer biologist between like 2005 and 2016, average price for a marten pelt from New York is probably about $40 to $50 on average. But sometimes you get I'd hear trappers say, "Oh, I got $90 or $100 for that marten pelt." So it just depends on the fur buyer and where the market is in any given year.
Okay. Gary noted that the reintroduction of fishers into the Catskills a few decades ago was very successful and wants to know if there are plans to reintroduce martens to the Catskills.
Yeah, so I'm actually working right now with my fur bear biologist, the person that I hired to replace me, and we're working on a American marten management plan for New York State. And so part of that is looking at other areas of the state where we could potentially reintroduce martens. Now, I've kind of already done that work for the Tug Hill, as you saw, and what we found there, Gary, is that again, the Tug Hill really didn't have any suitable marten habitat, likely as a function of the other carnivores that are present there. Now, the Catskills could potentially be a different matter, and that's something where I still need to take that resource selection function that I developed for the Adirondacks and essentially apply it to the Catskills. And it will be an extrapolation because obviously we didn't use any data from the Catskills to build the model, but that's something that that we plan to do and look at to see if there's any potentially suitable habitat in the Catskills. If it's there, it's going to occur at the very highest elevations, and a lot of the I would say probably anything below 3,000 feet, 2,000 feet, somewhere in that neighborhood, is going to be unsuitable habitat. So what I think the model is going to show, this is my prediction, is that there are small islands of suitable marten habitat in the Catskills, but it's going to be fragmented, it's going to be small, and ultimately it's going to be isolated from other populations as well. But I still think it's worth looking at.
Okay. At one point, you were talking through the different types of mast species that triggered cyclical responses of the rodent populations, and the question was whether hop hornbeam, eastern hop hornbeam, or genus Australia would similarly have. Did you any evidence that those cycle through mast years?
It does seem that when you start paying attention to trees that produce seeds, cones, nuts, what you find is that in a big mast year, this is, again, I'm speaking generally, but in a big mast year, it's almost like everything is producing. So apples and mountain ash and beech and hop hornseed. The cones don't generally follow that as closely. And then the other species of tree that I didn't really mention that doesn't follow it closely are the oaks. The oaks seem to be on their own cycles that are very, very strange and have a lot of variation and very hard to predict. But excluding those species, yeah, I do see. I mean, I see even around where I live in the Adirondacks, where if it's a big mast year, I typically do see hop hornbeam produce. I don't, just offhand, I don't know if that is, an important food source for small mammals, but I think pretty much any kind of seed or fruit or nut is utilized by small mammals, so it wouldn't surprise me.
Okay. Are grouse and turkey significant competitors for mast, either, I guess, for the relative to the rodents?
I don't know, but my guess would be no, just because, when these mass crops produce, it's massive. I mean, it essentially overwhelms the, what's called seed predation of all these different species that utilize this food source. It overwhelms them. And so with these mass cycles, we do see sometimes some years when that mass production is kind of at an intermediate level, and it can be very kind of like spatially clustered. So you might have like a beech stand, for example, that produces a lot of beech nuts, and then you might go over the next ridge and find another really nice stand of beech, and there's no beech nuts. So there is, and that even happens in big mast years. You do have interstand variability, but by and large, when they produce in such quantities that it literally overwhelms the wildlife species that are dependent upon them. And I can remember one year we were doing some work putting up camera traps, and it was a big mast year, and there were so many beech nuts on the forest floor that I just stopped, and I was just like gathering them and eating them because there was just so many. Like within a minute period, I had like two handfuls just full of beech nuts. So pretty amazing to witness. And so keep an eye out, look up into the canopy, and you'll be amazed in these big mast years of how many nuts are on these trees, and then eventually will fall onto the forest floor.
Okay. So next question. You described this fascinating relationship between mass production, the small mammal consumers of the mast, and then the carnivore consumer of the small mammals. Does that pattern, cyclic pattern, hold for other um, predator prey scenarios? And the example that was thrown out, and maybe I shouldn't give it because I don't want to kind of steer that, but it's like you got like squirrels and deer, and then predators like bobcats and coyotes.
Well, I'm just trying to think. I mean, certainly, I mean, as far as is the question, Pete, related to specifically the mass cycles or just predator prey
So
interactions in general?
so I'll read it. Do other mammals and birds have the same cyclic response to mast production?
Oh, okay. Um, so I don't have a lot of information on bird responses, but bears are another species that very closely track these cycles. And so we see very similar patterns with bear harvests, and then also when what happens is in years when mast mast produces in the fall, and then you have higher survival of cubs overwintering, and then in that next year, and the other thing I didn't mention too, I'm kind of tied in with this, is that there's also a relationship between what we see with the hard mast and the soft mast species. So what happens is if you get the perfect storm with bears, where you have a hard mass crop in the fall, and then the next summer is a dry summer, and the soft mass doesn't produce. And again, these weather events are correlated, but if that perfect storm happens, you end up with a lot of nuisance bear issues, and these bears are moving all over the place, and they're getting themselves in trouble. And again, that's something that we have seen here in New York.
Okay. Does the quality of the pelts differ between high mast and mast failure years?
Not that I'm aware of. I'm not sure, what that linkage would be. I mean, obviously, you might think about an animal that's in poor nutritional status, poor body condition, and that being reflected in the quality of their pelts. I haven't really seen that. We do have, I mean, it does get to the point, again, because one of the things you have to realize with all this that I'm talking about, at least in the study area I'm working in the Adirondacks, is that it's a low productivity system, because the forests are mature. Most of our forest stands are mature second growth or old growth. So primarily, greater than 100 years old, especially where martens exist in the Adirondacks. And so hare populations are really low to non-existent in some areas. Deer populations are very low. So really the primary prey that's available to martens during that limiting time of year in the winter are those small mammals. So we've documented in our work that when you have a mast failure and you start getting towards late winter, we've had starvation actually, occur quite frequently with our animals. And so I've handled, and I am circling back to the question on this, but I have handled animals that we've radio collared that, you look at them in the trap and they look, oh, this is a healthy marten. Fur looks good, looks fairly well fleshed. And then you drug the animal and you take it out of the trap and you realize that they're just skin and bones. So I have handled quite a few martens that were in really poor physical condition, but yeah, I didn't really notice any kind of differences in their fur.
Okay. How much did the GPS collars weigh?
So
It was
the
version 3, I think, that you ended up using.
Yep. So the version 3, actually I used the version 2 and the version 3. The version 2 collars weighed, I want to say about 34 grams, somewhere in that neighborhood. And that version 3 collar, the male, that one actually weighed a little bit more. That was more on the order of about 37 grams. And the reason for that was I used a thicker collar material, which I won't use again. And if you noticed on that, you may not have noticed it, but the female collar to the left uses a thinner collar material, and that's it's a green color. But in the future, when we're constructing the male collars, we'll use that lighter collar material. But yeah, 34 to 38 grams. And then that female collar is, trying to think here, I believe that female collar is about 24, 25 grams. So that's right on the edge. Like many of the females that we catch here in the Adirondacks are really small. So it's not uncommon to catch females that are 450 grams, 500 grams, 550 grams. So you're right on the edge as far as using that size collar.
So what's, for other people may not struggle with grams, what can you give me a context of something that weighs 30 grams, like a quarter, or
Oh boy.
an egg, a chicken egg, or
Well, what I will tell you, I can't do that, but what I can tell you is that 454 grams is a pound.
all right.
So you could do the conversion.
So it's 10% of a pound, so it's about an ounce.
So the
real quick math.
Yeah, but for the male collars, yeah.
Yeah. Okay.
Roughly 10%.
Okay. I can Google will help me with that one. So okay.
Yep.
Let's keep going here. Who was the person in Maine that you mentioned regarding timber management for marten? Oh, that was answered. Dan Harrison, University of Maine.
Yep, Dan.
Okay.
I really, as far as the northeastern U.S., I would say Dan and his students are really the experts on that topic. And it makes sense. I mean, the timber harvesting that occurs in Maine, and the importance of that to Maine's economy, coupled with the fact of desiring to conserve marten populations. And so he's done a lot of really groundbreaking research.
Okay. Owen's reporting that he saw a marten in southern Madison County, but he thinks that that's probably outside the range. Is that right?
That is, I mean, I'm old enough to say that would never occur because I've been surprised before on things. And we had, we had two occurrences of martens that were way outside of the range. Both of them were on the western side towards the Tug Hill, and one was killed by a car in the city of Rome, and the other one was um, incidentally trapped by a fisher trapper, and that was north of Oneida Lake, I believe, in the town of Amboy. So, is it possible? I would say yeah. Um, you know, probably not, just because it really would have to navigate a lot of roads and a lot of areas that are unsuitable. Open areas, exposing itself to predators, as well, avian predators, as well as other terrestrial predators that would be a lot more abundant there than what they are in the Adirondacks. But it could be, I can't, I can't confirm it and I can't deny it.
Okay.
And that's my story when we talk about mountain lions. I can't confirm it and I can't deny it. Um, so what's interesting though is I know my answers are longer than maybe than what people want, but one of the interesting things that we see is that when in these mass failure years that we've been talking about, that's when we see these really long distance dispersal movements. And so there's definitely sort of like this bottom up influence on dispersal, and it's something I want to try to squeeze from my existing data set if I can. And so if that's really when you're going to have those long distance movements, is when the mass fails. So
Neat. Well, Paul, I think you have answered all of the questions, and you've done so splendidly. And there are lots of people saying what a very great job you did. So I'll echo that. And I want to thank everybody. We've had 41 people stick it out here to the end, and that's a tremendous show of support and interest for a really fun topic. So I appreciate your effort in putting this together, and thank all the participants. So
yeah.
Paul, have a great evening. Happy St. Patrick's Day, everybody. You've got time for some green beer or green jello, whatever your pleasure. So, all right.
Great. Thanks,
Thanks,
Pete.
everyone.
Thanks, everyone. Have a good evening.
Thank you. You too.